Okay, so I'm just going to introduce Dr. Jim Wilson. It is a great honor for all of us to have Dr. Jim Wilson here to present our keynote lecture. He is, I'll just give a really brief story about how we met. But basically what happened was he is on email late at night because I was reading a Google alert, and I was always looking for the pioneer in gene therapy and how we can bring this to children with Angelman syndrome. And I got a Google alert, and it was an article about Dr. Wilson. And I decided to Google him and see if I could find his email. And it turned out he was at the University of Pennsylvania. And I did my residency at the University of Pennsylvania, so I felt like there was a connection. So I was going to email this guy and see if he would ever write me back. And I wrote him, I don't know if you remember, at about 11 p.m. on a Sunday evening. At about 12:30, I had a response. I kid you not. And the response was, let's chat. And we chatted, myself and Paula and Sharon, and we had a meeting, and we ultimately had a phone conversation, which became an in-person conversation. And I'd like him to share with you ultimately how that went, but obviously it went pretty well, because he doesn't take on many genetic disorders, and it didn't seem that it was that hard to convince the gene therapy program in the Orphan Disease Center to realize the potential for Angelman syndrome. So that was a really exciting moment, and because of that, we have obviously invested quite a lot emotionally and financially into this program. So we're very excited to hear what he has to say about that. But what you also need to know is that Dr. Wilson is one of, if not the pioneer in developing viral vectors for the delivery of gene therapies. So all of the vectors that are currently, or most of the vectors that are currently being used in clinical trials, the adeno-associated virus, that was him. So we are really lucky to have him in the room with us today. He knows this better than most. He knows what to expect. He has a grand amount of experience, and I think that we really can use what he has learned over the past 10, 15, 20 years to really help our kids. So he is a physician at the University of Pennsylvania, also a scientist, and he is the director of both the gene therapy program and the Orphan Disease Center. And he is really here to show what is possible for Angelman syndrome. So Dr. Wilson.
Great, thank you. It's a good thing I said yes, because if I said no, it would have been the wrong answer, obviously. A true force of nature here. It's an incredible pleasure to be with you today and to begin to join your family. As Allison had said, I've sort of dedicated my life to developing treatments for rare diseases and began to think about gene therapy actually 35 years ago. So that's been my dream, to make this a reality. And when we were approached by Paul and Allison about considering Angelman's, we put a lot of thought into this because we wanted to make sure that if we stepped to the plate that there was a reasonable chance of delivery. We run the Orphan Disease Center, and as you probably know, there are 7,000 rare diseases that fall under our remit. We commit to probably 10 or 12. And what we decided is there was a reasonable chance of success. It wasn't going to be easy. We could leverage our experiences with easier diseases. And ultimately, and again, this is only an hypothesis, that if we did this right, that there would be a chance for those affected by Angelman would actually regain function and improve the quality of their life. That's the hypothesis. So once we got over that hurdle, we then began to work together with FAST to develop a grant and to proceed forward with our research. But I thought for today what would be useful, since I suspect this won't be the last time that I meet with you, to give you background as to how we got to where we are in gene therapy. And then that'll give you some context to determine whether you think there's a chance of success or not. And I think my goal today is really about educating you about the field and what's happened so far, because you read about it in the news. And I could say that after 35 years of pursuing this dream, it's absolutely amazing what's happened over the last 6 months in gene therapy with finally approved, FDA-approved clinical trials in some diseases for which there weren't any other treatments. So that's the goal for today, and I hope I can do this in a way in which I can teach you at the end of the day. I am a professor, and I'm going to probably seem somewhat professorial here as I teach you. I will promise you there won't be a quiz at the end of this. So the students and the children probably are fine with that. I don't know about the adults. Want to just note that as a result of our work that we've worked with a number of biopharmaceutical companies and many biotech companies that we actually started that helped us move this along. So what's the key to success? Something that I've been pursuing for 3 decades. The key to success is delivery. What do I mean by that? How do we get the normal version of the gene into the right cells of the patient in a way in which it would be efficient and safe? And that's really what I've tried to pursue over that time, and that's where the notion of viruses became possible. So the problem with the gene, no matter what gene we're talking about, it's big. It's much larger than a small molecule, a pill, aspirin, penicillin. You can't swallow it. And even if you injected it, it would just float around and be degraded. So we have to package this in a way, in sort of a vehicle that allows the gene, when injected or inhaled, or however it's administered, that it circulates around the body, finds the right cell, targets the cell, and delivers its payload into the cell, not only the cell, but the nucleus, in a way that we reprogram the expression of that gene. And for diseases that are caused by an absence of a gene, what we're simply doing is we're replacing or introducing into the cell a normal version of the gene that is defective. So we're not actually going in and engineering or fixing the mutation. That's why they call it gene replacement. We're replacing the gene. So the key here to our current success was the notion of using a virus to deliver a gene. Now, viruses have their own genes, and as we all know, they evolved not necessarily to be our friends. And they have a series of pathogenic genes that, together of which, allow the virus to survive at our expense. So what we've tried to do is engineer the virus and its genome to take out the disease-causing gene and to incorporate the therapeutic gene, to attenuate it. And that's a fine line, and that's something that we've tried to straddle for many decades, and I must say with a lot of false starts and a lot of failures over a long period of time. So the virus that we decided to focus on right around 1990 or so is something called adeno-associated virus, or AAV. You've never heard of this virus because we don't even know if it causes a human disease. This virus was actually discovered in some sort of, as a contaminant of a laboratory prep of another virus. That virus was called adenovirus. So it could cause some disease in its normal form, but we don't know that. But it had certain properties that when we could engineer it, that at the very base, which is where we start, it seemed to be as safe as anything that we had studied up till then. And so there were a family of these viruses that were discovered in the 60s, actually. And we went into the archives and we pulled them out of the freezers and made vectors out of them. And the one that emerged as the first one that was tested was something called AAV2. So if we think about what we want to achieve in gene therapy for a genetic disease, we want to be able to inject the gene, we want to be able to program a cell to express a protein, and importantly, we would like that to last for the life of the patient. So how long would a genetically engineered cell in which an AAV introduced a gene last? How long would it express? Well, I'm going to share with you kind of a weird experiment I've been criticized for doing this experiment, but it still taught us a lot. This was an experiment where we took a gene for a hormone called erythropoietin. Erythropoietin, you may or may not know, controls the level of our red blood cell production. So when you can treat anemia with this. Normally produced by the kidney, but we put that gene into an AAV2 vector. We structured it so that it would express in any cell, and we injected it right into the muscle, like you get a muscle injection, of a monkey. And we actually asked the question, will those muscle cells express that hormone, and how long would it last? And so what you see here is the expression of this EPO gene, which is on the y-axis, over time. And the level of expression that we achieved was very high, which is good. We engineered the muscle cells to express the protein. But most importantly, it lasted for a long time. So this continued to crank out the gene product for 11 years without decreasing. So this suggests that maybe one could consider an injection of a vector that could last at least 11 years and hopefully longer than that. So far, good news. But we have a real delivery problem when we try to treat real genetic diseases. So there are two examples that I want to share with you where we've overcome the delivery problem. And I'll give you a heads up. We think we're on our way there in terms of, can we see that? I think I've got some loose connections here.
Oh, wait. Okay. Why don't we just take questions while they're figuring this out so that I think you'll get the most out of the slides and not just Dr. Wilson speaking. So does any parents, I know there were a lot of questions after my talk that we didn't get to. Are there any questions now? Yes? Why am I surprised? No, just kidding. I'm just kidding. Do we have a runner?
Sorry, I'm not trying to be annoying by asking a lot of questions. This is my first weekend and all this, so this is probably more actually for Dr. Wilson, but in terms of using a virus to deliver the gene or the protein, if the child already has the gene, so non-deletion, does that make the delivery of the protein a better option? Or is there any downside to delivering a gene that already exists but just isn't functioning? You know what I mean?
Yeah. So different ways in which you could net out no protein. One would be a deletion, one would be a mutation that renders the protein inactive. Gene replacement therapies bypasses both of those mechanisms because it essentially inserts the normal version of the gene in the context of the abnormal version. So if the abnormal version is making defective protein, it's essentially non-functional. So there would be no advantage of protein replacement or not.
Sorry, I wasn't sure if there's conflict in delivering a gene that may already exist. So
There should not be.
It's non-functional. It doesn't matter if it's present. It's non-functional. So you just need a functional copy, however it is, protein or gene. Are we up? No. Any other questions?
Back there?
Over here.
Mic over here.
I'm over here.
Yeah, hi.
Hi. So first of all, thank you so much for all you're doing for our families and our kids. Thank you, first and foremost. Second, it's a two-part question. So what happens to the virus vector once it transmits the UBE3A where it needs to be? So I'm looking at kind of like as maybe like a truck that has something on it, takes it somewhere, drops it off, then what happens to that? Second part of my question, so we all have different deletions or UBE3A, whatever genotype. For instance, Maverick has 4.96 deletion. Does the deletion size matter as far as the UBE3A that will be reintroduced or replaced?
So for your second question as far as a deletion versus a mutation versus a UPD versus an imprinting center defect. The gene replacement or protein replacement is only replacing the UBE3A gene, and it's not actually the whole gene. It's a part of the gene. It's different isoforms of that gene, and the gene is so big it doesn't fit in the virus. So you have to pick the most important part of that gene, because there's different forms of the gene in that whole segment of UBE3A. So it can't fit the whole deletion size. So the deletion kids would get replaced the part of the UBE3A gene that we believe is the most functional for humans, and the other parts that are missing would not be replaced. So for a mutation child, they're only going to be getting, they're only missing UBE3A, they're going to get that back. For a deletion child, they're still not going to have back the other parts that are
We
missing.
can't really hear you, or I can't.
Is that better? Yeah, just let me hold on to this. So for the deletion kids, they would be getting back UBE3A, not the other parts that are missing. For mutation kids or UPD kids or imprinting center defect kids, they're really only missing, that we know of, the UBE3A gene. So the gene replacement therapy is not multiple genes being replaced, it's one single gene being replaced, if that makes sense. So it doesn't matter, ultimately we're not changing it based on your kid or my kid or someone else's kid. It's the same gene for all of the children regardless of their genotype, which may have an impact on the benefit for certain kids compared to other kids.
Okay, so you're saying potentially somebody with UBE3A or UPD might benefit more, and potentially
theoretically,
than a deletion positive child.
Theoretically,
for
that
this
is
type of
that
therapy.
is a theoretical possibility. It doesn't mean that the GABA genes that are missing in the deletion kids aren't going to have major impact by OV101.
Okay.
And so it's going to be multimodality. They're still maybe going to need, various different types of anti-epileptic drugs, because the GABA genes they're missing are probably why our kids have a higher seizure phenotype.
Okay.
So yeah, there's going to be multimodality, but as far as the effect UBE3A has on our kids, that will be the same for all of us. We will all get that back. It's the other parts that may still need some work in other areas.
Okay,
if that
great.
helps.
Thank you so
I think
much.
we're back.
Thanks.
We're back? Okay.
All right, thank you.
You're good. You're on.
All right. Thank you to the guys back there for bringing us back online. Let's hope we stay there. So we left off where I was talking about two different models where we have overcome the delivery challenge, and one is treating inherited forms of blindness in which children are born without sight. And that's due to a genetic defect that renders a protein inactive. So we could potentially treat these diseases by, oh, by the way, whoever's taking care of the time, I do get an adjustment, right? Okay, all right. It's pretty aggressive down here. So how do we get the gene into the retina? The way we overcame this was a surgeon would insert a needle and inject a bubble of solution, as shown here, under the retina. And this just shows what happens when the gene gets into that space, and it actually creates a retinal detachment. But, as shown here, we've been able to demonstrate in animals that we could engineer the cells, and in this case, this was an experimental model that turned the cells green. So I want to share with you a journey that will lead to the first approved gene therapy study product for genetic disease in the United States. We expect this to happen around first week of January. This is a colleague of mine, Gene Bennett, whose dream was to treat patients with inherited forms of blindness. And soon after I arrived at Penn in '93, I began to work with her using viruses. And we went through a variety of different viruses and ended up with adeno-associated virus. But it started with a mouse over here in '94. We went to a monkey in '99, and then we had to show it actually worked in an animal model. And fortunately, in this disease, there was a dog that was born with the same kind of genetic defect that was treated. And this is one of the dogs within this colony, whose name is Lancelot, as you can see here in front of the U.S. Capitol. And Lancelot has testified before Congress on behalf of NIH funding. And apparently the congressmen are quite smitten with the dog. I don't know what that means, but But seriously, then she moved this in a very bold way into the clinic in children that were born with blindness, and I'll show you some data. And I knew this became real when one night I was watching America's Got Talent. I don't know if you remember, but there was one of the contestants, and he had participated in this gene therapy trial. So I'm going to show you a movie of a patient trying to traverse an obstacle course before gene therapy. This is a young woman trying to follow the arrows and clearly struggling. That's Jean in the back, nervously kind of watching her struggle. Bumped into the bucket, stepped on that obstacle, and basically didn't see the blue one. And so there was this is after gene therapy. And what we've learned from this study was really incredible, because these patients had not seen perceived light for years, decades. And we weren't sure when you turn the switch on, would their brain recognize it? And clearly, this human experiment showed that. So we hope that you'll be reading about this being the first FDA-approved gene therapy study for genetic disease very soon, clearly changing the quality of these patients' lives. Another interesting story that I want to bring up, because I think I'm going to be asked about it. I saw the questions in advance. And this was the first gene therapy approved study in the West, and this was in Europe, an agency called EMA. And it happened to be our vector that we developed for this. And it's a disease that causes a very high level of triglycerides in the blood. And as a result of that, their blood looks milky, as you can see down there on the lower right. That's not normal looking blood. It should be clear, absent the red blood cells. And because of this, they develop severe episodes of pancreatitis and die. So they're missing a defective gene that's expressed from the muscle. That's an enzyme called lipoprotein lipase. So gene therapy is to take the gene, put it in one of our vectors, inject the muscle, program the muscle to express the enzyme, and hopefully persist for a while. And this was a vector developed by a postdoc of mine, Weidong Zhao, and it was called AAV-1. It was part of those 6 AAVs that we pulled out of the freezers. Importantly, we were able to show, or it was not us, but other scientists that we licensed the gene to, the vector to, show that in a cat model, again, a large animal model that had this disease, that they could effectively clear the serum of this milkiness, which means their triglycerides are down. It was a challenging study, required a lot of muscle injections, but it was a pioneering effort. And in 2012, the first approved gene therapy product in the West, in a product called Berra. And there the story got a little interesting, because the company decided that they were going to charge $1 million for the injection. And they successfully sold 1 dose to 1 patient, and then after 5 years of no one basically using this, they pulled it from the market. Now, to be fair, that this product was a pretty weak product. It really didn't cure the disease, and there was questioned whether there was efficacy or not. But there is a brewing discussion and potential controversy of how do you price a cure. And I know I've been asked about this by members of the community here, and be happy to share some ideas about that later on if you'd like. So right around the turn of the century, the field had suffered a number of adverse events, including some at the University of Pennsylvania. And the technology that we had at the time either was unsafe or didn't work very well. So AAV 1 through 6 really wasn't adequate for other organs such as liver and heart and the central nervous system, which is where we wanted to end up. So we spent a lot of time studying viruses. I've become a virologist, and we learned that these AAVs that came out of these laboratory dishes a long time ago actually are are infectious agents in all of us, and we all have remnants of these AAVs following some kind of infection that we had. Interesting, almost bizarre, concerning observation, but the fact is that we were able to reclaim remnants of these viruses and create a whole new portfolio of viruses that we developed vectors around and develop them so they were more efficient in other organs. I'm going to share with you really two ideas ultimately leading to where we are with Angelman's. So the second kind of crucible in which we evaluate gene therapy, where the delivery problem is easy, is the liver. Now, the liver is a big organ, and how do you get genes into the liver? There's something very different about the liver, unlike any other organ, and very different than the brain, and this is why it's important to understand, is that the liver, the blood vessels of the liver have large pores or holes in them. And there's a function to that. But no other capillaries or blood vessels in our body have these pores. What this means for us is we can inject a vector into the blood, and it'll get across that barrier into the cells on the other side, which are the liver cells. Without these pores, the vector will just circulate in the blood, and it really does no good. So again, this is the delivery challenge that we overcame just by taking advantage of basic anatomy. So the disease that was initially tested and is aggressively being developed for one of our new vectors called AAV8 is hemophilia. And many of you may know males who have hemophilia. It's X-linked recessive, affecting only males. And these boys are born without the ability to clot. So if they suffer a laceration or a mild trauma, they could develop a life-threatening bleed or a spontaneous bleed, such as a bleed in the head. And without treatment, these boys would die. There is a treatment. It's called protein replacement. And in this case, protein replacement is manifested by the patient coming to the hospital, or it could be done at home, receiving an infusion of a protein in the blood that leads to an elevation in the serum protein. And here's a young boy administering his treatment. This occurs 3 times a week for the life of children with this disease. So there clearly is a burden of treatment, but it's potentially treatable. So in the upper left-hand corner, what we've plotted in a schematic way is the serum or the blood levels of the protein that's infused. So you see it goes up, then it comes down, because proteins don't last forever. They don't have a way to self-perpetuate themselves or to express themselves. And so then you have to infuse it again, infuse it again, infuse it again. Turns out the cost of this treatment that saves the lives of these patients is about $300,000 a year for the life of the patient. So what we did, is shown in the upper right, is we took the gene encoding that protein, put it in one of our vectors, it's called AAV8, injected it into the blood, got across that barrier, got to the liver cells, engineered the liver cells to express the protein, and hopefully do this in a very stable way, like that monkey that we injected with that EPO vector. And schematically on the upper right, we would expect a one-time infusion would lead to a permanent production of the factor. And over in the lower left, a dog model came to the rescue here, and these are dogs born with this disease, and we get referrals of these dogs frequently, actually, in which the dogs develop life-threatening bleeds, and we put the normal version of the dog blood clotting factor gene into a vector, infused it into the blood, did it with an AAV2 vector, didn't work, discovered our new vector AAV8, and you can see that the levels of the protein are high, and they persisted for the life of the dog, and the dog died of natural causes at 12 years of age. So with that as encouraging data, this was moved into the clinic in patients with hemophilia B in the lower right, and again, these are blood levels of the factor following a one-time infusion. And basically, in many of these patients, although still in early clinical trials, this circumvents the need for their protein replacement. So it's a one-and-done, one-time genetic cure of hemophilia. And there are 8 companies at least trying to chase down gene therapy for hemophilia right now. But we're talking about Angelman's, which is a neurologic disease. And we've got a challenge, which is a barrier, which is called the blood-brain barrier. So when you inject something into the blood, it doesn't easily end up on the other side of the blood vessels in the brain, where you need to deliver your small molecule, your protein, or your gene. And so how do you overcome this barrier? We're going to share with you ways in which we've approached it, but there's an additional problem that you need to understand, and that's how do we model this for the treatment of humans? So what we've depicted here, they're actually pictures to scale relatively of the brains of different animal models that we use in our research: the mouse, the rat, the cat, the dog, macaque is a monkey, and then humans. So it's not only a challenge of getting the vector there, but this challenge is greater the larger the organ is that you need to target. So most of our studies start in mice. Now look at the size of the mouse brain versus a human brain. I can suggest to you it's a lot easier to accomplish this in a mouse than it would be in a human. But it's a good place to start. It shows the concept. But basically what I've dedicated most of our work to over the last few decades is how do we go from a successful mouse experiment to a successful human experiment? And that always isn't quite easy. And this creates a certain challenge in terms of delivery, right? That's the challenge. So we've approached it two different ways. Aspirationally, we would like to develop a vector that when infused into the blood, got across the blood-brain barrier and delivered its payload to all of the genes in the central nervous system that you want to deliver it to. It's intravenous. And we actually discovered a vector called AAV9 that is being used extensively in clinical trials, as Allison had said, for treating neurologic disease, that shows some level of sort of success in getting across this blood-brain barrier, which is really amazing. We still don't understand why. But as I'll show you in a minute, has demonstrated incredible, almost miraculous success in a disease. But we feel that a more efficient and safer and effective way is to inject the vector into this fluid that bathes your brain and your spinal cord called the cerebral spinal fluid. And some of you, unfortunately, may have been involved in a procedure where a needle is inserted into your lumbar space to access fluid, a lumbar puncture. Or you can insert a needle into reservoirs that are around your brain in which it's more effective. So in that case, it would be a one-time infusion. The needle comes out. The vector would bathe the central nervous system and hopefully deliver more efficiently vector genomes. So how well does this work? Done a lot of work with AAV9 and other vectors, but AAV9 in particular, in comparing a lumbar puncture to an injection in the cervical spine, which would be right here, or an intravenous approach. And in anyone other than infants, the intravenous doesn't work. And even when you inject it intravenously, it's relatively unsafe. So we're really, for this program and most of our programs, destined to an injection into the cerebral spinal fluid. And this shows pictures of a monkey that received a vector through one of these injections. There's a cortex, there's the spinal cord, the hippocampus, and the cerebellum. And these are different regions of the brain that may or may not be relevant to different diseases. But in many diseases, you need a broad distribution of genes. So wherever there's green, that means the cell has taken up the gene. And the most efficient target for an intravenous infusion of vector are cells of the spinal cord, the motor neurons. So how do we plan to roll out gene therapy, and where does Angelman sit? So I've thinking about this talk, I've sort of put on paper what we've been doing without putting it on paper first, so it helped me develop some discipline around this, and thank you for that. So there are three different levels, the easier to the harder. The easier would be to focus on diseases in which the pathology is restricted just to a limited number of cells localized to a particular area of the central nervous system that we can get vectors to. Maybe that's self-evident. And I'll show you an example of that. The next level would be diseases in which there's more global pathology, possibly related to Angelman's, where you have to target many cells distributed throughout the brain. And that's going to be harder. Remember delivery. But it could be easier if you didn't need to get the gene into all of those cells. So global disease, but efficacy with a subset. And I'll show you an example of where we've succeeded with that. And the heaviest, hardest challenge would be a disease where we had to get the gene in every neuron of the central nervous system in 100% of the cells. So we'll talk about success in 1 and 2, and then we'll think about where does Angelman sit in this paradigm. So the first is, we call it the low-lying fruit. So this is a disease. It's called spinal muscular atrophy type 1. It's a disease that affects the motor neurons of the spinal cord. Those are the neurons that emerge from the cord to the muscle that innervate the muscle. These degenerate in different diseases. Probably the one you're most familiar with is ALS, where patients develop progressive muscular weakness and die. But there's a very severe early onset form called spinal muscular atrophy that's due to a specific genetic defect in which these babies are born normally, but they're diagnosed before 6 months of age because they never reach a milestone, not a single motor milestone, never lift their head, never roll over, never crawl. And most die by the time they're 12 to 18 months of age by a progressive loss of their motor neurons because of a genetic defect. So we know what the cells are, which is important. They're the motor neurons of the spinal cord. It turns out those are the cells we can get vector to pretty easily. So far, it's lining up as maybe a first proof of concept. How about the gene? It's a pretty typical loss-of-function recessive disease in which there are 2 different forms of relevant genes in our bodies called SMN1 and SMN2. SMN1, when genetically defective, both parents, you inherit a defective gene, there's no SMN1 activity. And it could be a variety of different genes, speaking to the question that came up before it. Gene defects, it really doesn't matter because we add in a new version of SMN1. So what were the preclinical data that supported moving into the clinic for spinal muscular atrophy? Well, there were 2. One is, can we get the gene, do we know what the cells are, and can we get the gene to those cells? So on the left, that's just a picture of the part of the spinal cord in a monkey that we injected a vector to, and you can see they're all staining those large orange bodies, and that's all of the motor neurons. We then licensed this to a company called Avexis, who together with colleagues at Nationwide Children's in Ohio, tested this in a mouse model that on the right, when you look at the green, that's a survival plot of animals without gene therapy, and they all died within 3 weeks. But then with high enough dose AAV9 vector expressing SMN1, targeting the motor neurons, there was 100% survival. So just 2 weeks ago, the seminal paper describing the first clinical trial of gene therapy with AAV9 for SMA was published in the New England Journal of Medicine, and I just want to share with you the data. So what we have here are 2 cohorts. Cohort 1 were SMA type 1 patients who received a vector at low dose, and what is plotted here is when they got vector over time, and importantly, where there's a circle, that would be an event, a bad event that they need a respirator in order to breathe, because that's usually what leads to their death. And then in cohort 2 were 12 subjects diagnosed before 6 months of age that received gene therapy at the higher dose. And at the bottom, you see their time of following birth. I guess all I can say is all of these patients should be dead, but they're all alive. And they're still alive, with only one that had an event, and that was at the low dose. There are different ways to measure motor function, clinical scores, composite scores, and this is called the CHOP Intent score. If you look in cohort 2, low score means not a lot of muscular function. Following gene therapy, the scores actually went up, where historically they should stay flat because they don't gain any milestones. So there actually was an immediate regain in function in some of these patients. Now, this isn't curing the patients at all. It's prolonging their life, but a much better quality of life. So if you really look at what matters, which will be endpoints in their pivotal trial, are their motor function. So after gene therapy, 11 out of 12 can talk. 11 out of 12 achieved head control. 9 can roll over, and 2 are walking independently. So this is moving very rapidly into a phase 3 trial, and FDA actually may approve this product just based on these data right now. You can imagine, I try to imagine what that would mean to parents who are born with a patient with SMA type 1. Interestingly, Allison, when it starts to happen, it starts to happen. While this was just rolling out, Biogen with an ASO developed another treatment for SMA that was just approved in December. So now before these families had nothing, and now they have the ASO or the gene therapy, so it's a different dilemma. Gene therapy is not approved yet, but it will be soon. So I think it does represent sort of the momentum on multiple fronts that diseases such as SMA and hopefully Angelman will be seeing some progress. Now, that is the easiest of easy targets for gene therapy, I believe. Now we have to talk about diseases in which there's a more global disease affecting many parts of the brain, many cells of the brain. And so we've thought about that as sort of a next step, and this would be diseases that cause global pathology in the brain that can be treated by correcting only a subset of the affected cells, because we can't get it into all cells at this point with what we have. And there is a group of diseases where we think this could be actually true. They're called lysosomal storage diseases, or MPS diseases. And these are diseases, they're a family, all different genes in a pathway, that when they're defective, they lead to storage of abnormal byproducts in the cell, as illustrated here under the disease. And then the cells die, and they affect the heart, skeletal muscle, ocular, and importantly, central nervous system. And there are treatments for these diseases so that they don't die of heart disease and that they don't develop debilitating musculoskeletal disease. And it's enzyme replacement. It's a protein replacement. You fuse it in the blood, and it treats their disease, but it doesn't get to the brain because of the blood-brain barrier. There is one approach, which is bone marrow transplantation, and like the ALD model, this is effective in only a subset of patients, but it's a way to get genes into the central nervous system. So what about gene therapy? Well, gene therapy here would be to treat the storage disease in the central nervous system. So our proposal is to inject a vector into the cerebral spinal fluid, and we hope to get that vector into a subset of cells. Well, I know we will. And that will treat those cells, so the storage goes away. But the problem is the other cells are still diseased. But there's something very interesting and opportunistic about this particular family of diseases, that a corrected cell actually can cooperate with an adjacent cell and fix the adjacent cell. So you can get more bang for your buck. You can fix all of the cells by only correcting a few of the cells. It's something called a bystander effect. So we move forward pretty aggressively at trying to move this into the clinic. On the left just illustrates the pathology in patients with this disease. And what you see is that's a section of the brain, and all that brown staining is the storage defects in all the neurons. So this is a global disease affecting really major parts of the brain. So we have to clear those storage defects. What we've done, again, a dog model of this disease was available at the University of Pennsylvania in a disease called MPS 7. And what we show here, if you just look over on the very right, we injected the dog one time into the cerebral spinal fluid. We were able to achieve gene transfer into many of the cells, as you can see here, but not all the cells. This isn't all red, although it's a lot of red, and wherever there's red, there's gene, messenger RNA, protein. But those cells cooperated with other cells, and as shown here, the storage defect that is typical of this disease. So if you compare normal with MPS 7, that brown kind of deposition of stain is the storage defect. And then when you give the vector, which we call IC, despite the fact it doesn't get into all cells, can clear all of the storage defects. So AAV9 is in the clinic in a variety of diseases: spinal muscular atrophy. These are active clinical trials. A variety of storage diseases: Batten's disease, Sanfilippo, and also another disease that's a neuronal disease, giant axonal neuropathy. There are two other diseases in which AAV9 has been used under compassionate use: ALS and another storage disease. And there are an equal number that are IND or IND ready. And so you're going to see a huge effort moving from the clinic, from the bench to the bedside of CNS gene therapy. And what my hope is, and having the opportunity to work with you, because we're sort of got the pulse on all of this, is that we could benefit from this experience and draft off this experience and leverage this experience as we start to think about Angelman. So we're not doing it from scratch. So what are the prospects for gene therapy of Angelman syndrome? We've got a lot to learn yet to really get this right. And all the work that the scientists are doing in the lab, studying the mouse models, studying biomarkers, studying pathogenesis, feed into this. But there are some important questions. What are the target cells? How many cells? What cells do we need to get the gene into? What is the gene that we want to use? Now, the one rule in our lab is we don't want to do gene therapy with the wrong gene, let me tell you. But there are different forms of this protein, and we got to get the right gene. Is the level of expression important? Is too much expression could be bad? What are the animal models that we have available to us? And importantly, how are we going to measure success in the clinic? So this would be my hypothesis, is that Angelman would fall in the middle category, not the lower category. The lower category is the most challenging, which is you have to get the gene in all the cells in 100% of the cells. And our hope is that despite the fact this is a global disease, that we could achieve some level of benefit to your children using technologies that can target many but not all of the cells. And there just may be a threshold. And the other thing to start to consider as we think about and learn more about the disease, and that's why the science of the disease is so critical, is that we may initially succeed in treating a certain aspect of the disease, maybe not all aspects of the disease, with the goal of treating all aspects of the disease, and identifying what those priorities are so that we can walk before we run and be realistic about what our expectations are. But if we do it, my hope is it would be permanent. And we'll need, obviously, those that will be driving this, your input into that. So as we heard, there are different ways to try to treat this disease, but what we're talking about is gene replacement, getting back to that question. Taking a normal version of the UB3A gene, introducing it into a vector in a way it could be expressed in whatever cell we get it into, and then populating in cells of the central nervous system this normal version that makes the normal protein that then would correct the defect in those cells and potentially benefit other cells that don't get the gene. We do need to decide which form of the protein we need to put into our vector so that my graduate students don't get it wrong. We have to get the right gene. And so there's a lot of work and there's been a lot of discussion that we've benefited from your consortium, your community, to try to figure that out. And I think we're moving quickly in that direction. So where we are in our particular programs is we've incorporated into our first generation gene therapy strategy the best components of what we have available to us now, which is an AAV9-like vector. We've actually already started to make it a little better. We've injected mice at high doses of vector to achieve very high levels above background levels in mice, with the concern that may actually may cause toxicity. And all I can say is the initial readout is there was no toxicity, but we have to refine that question to make sure there aren't subtle abnormalities in the mouse. But so far, it looks like that may be an issue that won't limit our progress. We're now working in animal models with our first-generation vectors. And then really, I think, is why Allison and Paula came to me, is not to do 1 and 2, but to do 3. And that is to really incorporate into this program the best of the best. Not what we started to think about 20 years ago, but what's coming out of the laboratories now in terms of new vector development and learning from our other trials. I do want to share one sort of science-y data set because some of you may be familiar with this. It's about delivery, and if anyone can make an advance to broadly distribute vector to all of the cells of the brain, it'd be huge. And God bless them if they could. We've been trying and we're making some success. But there was a paper that came out just about a year ago that was based on a version of AAV9 called PHPB that was engineered and selected for something that worked better in a mouse. It was a certain kind of mouse. We call it, it's called a C57 black 6 mouse. And if you look over on the right, you look at AAV9, it says 1 × 10⁻². That doesn't look like a very green brain because that's a mouse brain with AAV9. But you look in the middle, AAV-PHBB, that's really green. I mean, this was a single intravenous infusion into this mouse that completely repopulated the entire central nervous system. Unfortunately, it's unique to that mouse. So we followed up on this and we learned that this is unique to the black 6 mouse. And when you study it in another mouse, such as a BALB/C mouse, it's no different than AAV9. And when you inject it into monkeys, it doesn't work at all. And in fact, we're experiencing severe toxicity in some of the monkeys. So the other aspect of what if you're in it, in gene therapy, you're in it, and you're going to be hearing a lot of stuff. I can promise you, on your behalf, we're going to chase them all down. They're going to go through monkey testing and large animal testing before we render any opinion about anything that would be worth coming forward from our lab or any other lab. I'm putting them on notice. So where are we? It was many, many years ago when I showed up at the University of Michigan for my first job. I was sharing my laboratories with a guy by the name of Francis Collins, who's a famous scientist, geneticist. He was studying CF, cystic fibrosis, and he asked me to get involved in gene therapy for CF. That was before they had cloned the gene. I said, well, that's going to be really hard, Francis. But turned out he had cloned the gene. He hadn't told me yet. It was published three months later. But then the community with gene therapy and the gene, it's going to happen. It's going to happen. But I said, we don't know what the CF gene does. We don't really know where it's expressed. But the hope was maybe we could cure CF, AS, without really understanding what the gene does. Because just put it back in. If you can get it in, then it should be helpful. That's possible, but probably not. We'll go forward with our best vectors, putting the genes into the animals and studying safety, and we're full on, but we have to keep investing in understanding the disease, understanding the gene in an interactive way. If that doesn't work, that kind of science would drive which cells, what subpopulation of cells, which would drive delivery. So what are the challenges broadly that I would say to the community? Define the target cell population, so that would assess our delivery challenges. If we only need a limited number of cells, this may be easier than I had thought. And also the goals for the clinical development plan. What parts of the brain are associated with what parts of the clinical manifestations of the disease? If it's the hippocampus, we can get to the hippocampus. If it's the cerebral cortex, harder. So far, nonspecific gene expression is not toxic, but obviously more work would be needed. And we're proceeding in parallel with translational studies and working with the community through the coordination of FAST to try to inform our approach. And we will loop in improvements in technology as we go along. I'm going to give you a heads up, though, that perfect can be the enemy of the good, and there will always be something better, and it's something better. And at some point, we're going to have to make a decision collectively about what is the best for what we have and move forward. And we've got a lot of work to go before we get there. But do appreciate, on behalf of our lab and the Orphan Disease Center, to be part of this journey with you. Thank you.
So we have time for a couple questions.
I have a couple of questions. One was earlier on in the presentation you talked about the gene therapy for SMA, and you said it wasn't a cure. And I'd like to know, why was it not a cure? Because it seemed like it was a cure.
Yeah. We get to them too late. But by the time they have symptoms, they've lost 90% of their motor neurons. With newborn screening, where you could intervene before symptoms, we think that that's going to be a very different outcome.
The next question I had was, there seemed to be some success over the past couple of years with ultrasound to bridge the blood-brain barrier for chemotherapy drugs, and I just wondered if that would be useful at all, or has anybody explored that for bridging the blood-brain barrier for viruses?
Yeah, I mean, people have done some work in that area. We think it's just easier to go intrathecal in the CSF. It's just one-time infusion, and you circumvent the more systemic distribution outside the brain of what you give. Not completely, but that's... Let me tell you one other issue, that if you go via the blood to get to the brain, 30% of us have antibodies against these vectors, and you won't be part of the trial. If you go into the thecal space, antibodies are irrelevant.
A couple of weeks ago, a guy in California received a gene treatment for Hunter syndrome from Sangamo Therapeutics using a zinc finger technology. I was wondering how that might affect Angelman, and if at all. And this is a disease of the liver.
It wouldn't. It basically was a way to, it was a form of gene therapy, injecting an AAV to introduce the normal version of that gene into the liver cells. And it got pressed because it actually introduced the gene into the chromosomal DNA using genome editing. But it's much less efficient than if you just would have done gene replacement. And I've told them they shouldn't have done the experiment, but they went ahead. I'm not a shareholder of the company, and I'm a professor who speaks his mind, so.
Oh, okay. Hi, that's
Hi.
great presentation. Y'all have so much amazing experience and success. It's really wonderful. I know there's a lot of questions among the families about what is the difference exactly between your program and the program at Agilis, and if you could explain that a little bit, that would be really great.
I'm going to learn more about the program at Agilis.
Yeah, let's save that for the roundtable, because
yeah.
Agilis is speaking next, and they'll share a lot more about their program, and then they'll all be on a panel to answer that question. That's one of the questions all the families wanted to know, so we'll get there, I promise. So you don't have to speak on behalf of Agilis.
Yeah.
Hi, thank you. I think we can all appreciate how quickly the science is moving, but as parents, it can't move quickly enough. And I know that this is, maybe impossible to answer, but what do you think of in terms of timeline for when this can get into human clinical trials? So
we have a lot of experience moving these therapies in clinical trials. That's why they came to us. We brought programs into 8 different clinical trials last year, various diseases. If you pretty much knew what you were doing, you knew what the target cells were, you knew what the gene was, you had a pretty good idea that you could get them into the cell. From that moment to the point where you'd have approval to do a trial, it would be about 18 months. Now, that's not where we are with Angelman. And so there needs to be a lot of work to try to sort that out. When we get there, then it'll be an 18-month trigger. And that becomes turnkey, almost, that 18 months. Because it's a platform. It's the same vector. It's the same manufacturing. It's the same FDA. But it's really, I believe, in the exploratory phase still, to try to determine whether, if we go forward in the clinic, that we're going forward with a best-in-class. And that's a hard, it's a difficult decision to make because of the pressure of time. My own view, and that's why I signed up, is let's make sure that we know what best-in-class is by leveraging all of these experiences, huddle and figure out whether it's time to move into the clinic or not. And that's a decision that's made by the community.
You said there was an instance where the PHPB had a toxicity effect on a monkey.
Mm-hmm.
You also said there was an instance where AAV9 had crossed the blood-brain barrier one time. If you were to inject it into the spinal fluid, is there any way that it could cross the blood-brain barrier the other way and create a toxicity issue?
So the systemic toxicity when you inject it into the blood is really what is, I think, most concerning, and because of the broader distribution of the vector. But it's dose-dependent, very dose-dependent, only at really high doses. So whatever leaks out when you put it into the thecal space is about 100-fold lower than what you would expect to see with toxicity. And there are ways in which we could, I think, completely avoid it as we're working on.
One more.
I just had a question about cost. You said earlier you could give some information on if this were to be available to parents. Like, what would the cost look like? Is it like $1 million, or is it affordable? And does insurance ever help cover any of these kinds of treatments, or is it so unknown right now?
Well, for rare diseases, whether it's a small molecule, Kalydeco for CF, or enzyme replacement therapy, they usually can recover what the companies charge, which is around the $300,000, $400,000, $500,000 a year for the rest of the life that insurance companies pay for, and not access like that throughout the globe. But there is high appetite for treatments that change the lives of patients with rare diseases. I'm not saying it's justified, et cetera, or whatever, but that's pretty much how it plays out. But then that company's collecting $300,000 for the rest of the life of the patient. So what if you just inject something and boom, you're done? Creates a different business challenge. We'll figure that one out if we can make it work. I promise you that one.
Thank you so much.
Yep.